Hey guys, this is Mr. Hyatt. This is part 3 of the Chapter 10 AP Biology lecture from 2017-2018 on photosynthesis. Sorry that we got interrupted by the bell in the last video. Excuse me. This is another version, another picture of the things that we talked about as far as chemiosmosis in the last lecture. Remember that we've got the thylakoid space that's the outside of the cell, and we've got the stroma that's the inside of the cell. And so we're pumping hydrogen out, it's diffusing back across, it's interacting with the photosystems, and we're creating ATP. So just again kind of a summary. There can be as much as a 3 pH difference between the thylakoid space and the stroma. Remember, pH is power of hydrogen, so there's a lot more hydrogen outside than there is inside when we're talking about a 3 pH difference. Remember, we're talking about the logarithmic scale here, so that's a lot more hydrogen. And there's a picture that we've used to kind of figure that out. And yeah, so just kind of a comment. That's why it's marked level C. Okay, so dark reactions are known as the Calvin cycle. We got a picture of Calvin over here. It's how plants actually make their carbohydrates, make their own energy. They don't require light directly, but they do require ATP. So kind of the idea, you got to spend money to make money. Got to spend energy to make energy. So synonyms, Calvin cycle or C3 photosynthesis. Don't be confused because there's also C4 photosynthesis. So, in the dark reaction, we're going to use ATP and NADPH. Interesting. We're using the outputs of the light reaction. So the outputs of one reaction become the inputs of the next reaction. That sounds like a chain reaction that's going to make something really big and really meaningful. Notice this happens again in the stroma. It's going to use the ATP and the NADPH from the light reactions to do something. There's a really important enzyme involved in this. Notice I called it level B. I probably should have called it level A because it is so important. But I'm just not going to test you heavily over it because you don't have the organic chemistry background yet to really truly understand how all this is happening. Once you get to organic chem and you can do electron diagrams and you can see how the electrons are moving around, that would be a more appropriate time to have that conversation. So Rubisco is short for this long thing, which if you're a strong scientist person, you ought to be able to break all those things down. Bisphosphate should mean something to you. Carboxyl should mean something to you. ACE should mean something to you. And ribulose, you should be able to piece together that os means something and ribu means something usually. So you should be able to piece together basically what that is. Essentially, it's an enzyme that that adds carbon dioxide to an acceptor molecule. I call it the most important enzyme on Earth because without photosynthesis, there wouldn't be any life on Earth. It's the base of our food chain. So if we look at the Calvin cycle diagram here, we've got our input of carbon dioxide. Remember that this whole process, photosynthesis, takes carbon dioxide out of the air. So we need to figure out how that happens. We're taking carbon dioxide in and we're fixing it. Remember that fixing means taking gaseous anything and turning it into a solid or a liquid form. In this case, it's a solid form. So we're taking CO2 and turning it into something else that holds carbon. In this case, we're turning it into 3-phosphoglycerate. Spending some energy. Energy investment phase. Excuse me. Okay, we're going to invest that ATP. We're going to invest that NADPH. So Rubisco changes into this 3-phosphoglycerate, and we spend a little bit of energy. We spend that NADPH to reduce this to this. Just a different molecule. Continue on the cycle. That feeds into this G3P that's going to excuse me, spend more energy, and we're going to get our Rubisco again. So there's our cycle of how we use and then recreate Rubisco. I intentionally skipped over this phase right here in reduction. It's the whole point. G3P is our sugar. We're going to combine those with glucose. Combine those to make glucose and other organic compounds. So here's where the energy comes from. Like I said, this is how plants make their quote-unquote food. Well, they're just going to package it and use it, just like we would in our mitochondria. They're going to use that ATP locally. Or if they do a lot of it, they've got some extra, they can store it in a leaf that a consumer can then come along and can consume and use as energy. So similar to our other slide on requirements and products, C3 or the Calvin cycle, we need carbon dioxide again, we need a bunch of ATP, and we need quite a bit of NADPH. And we're going to make sugar, ADP, and NADP plus. Now that sugar we're going to be able to break down and turn back into ATP, but that's a different process. That's called respiration. So here's kind of an overall summary. We've got photosystem 1, photosystem 2, and those electron transport chains that are going to take water and light and essentially make oxygen. So there's the waste part of the reaction. It's going to make ATP that's going to power this process, the Calvin cycle, where we produce sucrose, that's a sugar, and some side products, of course, and then we recreate Rubisco as well. So it's a cycle. Photorespiration is a bad thing. It's when Rubisco accepts oxygen instead of CO2 as a substrate. It's really, really similar to what happens to humans and other animals when we get carbon monoxide poisoning. We've got the wrong molecule being accepted, and then the process is going to continue, and it's going to mess things up. So as a result, we're going to make no ATP, which is kind of the point of the process, is to make ATP. And it decreases photosynthesis or photosynthetic output by as much as 50%. So it's obviously a pretty big deal. The thought is that might reflect a time when oxygen was less plentiful in the atmosphere. So it wasn't an evolutionary advantage to not be able to use oxygen. There wasn't enough oxygen around anyway, so there was just no competition. There was no reason to have that. But over time, oxygen built up, and there we do see photorespiration. Well, there are two strategies that have evolved to combat photorespiration. That's C4 photosynthesis. Remember, the Calvin cycle is called C3. So C4 is going to be a little bit different. And then there's CAM photosynthesis. So we'll look a little bit more at each of those here on the next few slides. Specifically C4, the biggest difference is that it's going to use a different enzyme. It's not going to use Rubisco. It's going to use PEP Carboxylase instead of Rubisco. So it's going to separate the CO2 capture from the carbon fixation to sugar. It's going to geographically move it apart. So that what was one step in C3 photosynthesis becomes C4 photosynthesis. Notice it's still going to use C3 to make sugar, but only in the bundle sheath cells. So here's what we're talking about. In these cells, we're going to make sugar. They can't receive light directly anyway. These mesophyll cells are not going to go through the C3 with the Calvin cycle. They're going to go through the beginnings of the light reaction. They're going to pass some of the materials, the raw materials, into these bundle sheath cells, and they're going to make sugars. So again, we've separated them geographically. Over here, we've got all the carbon fixation. We've got, essentially the carbon fixation. And over here, we've got the sugar generation piece of it. Like I said, we're going to use PEP Carboxylase instead of Rubisco. Sorry, lost my train of thought there. We can use carbon dioxide all the way down to 0 parts per million. So you can, even in an oxygen-rich environment, PEP Carboxylase is going to grab CO2 and not oxygen. So that's going to prevent photorespiration. We've got 19 different plant families that can do that. Sugarcane, Bermuda grass, and crabgrass are examples of those. So if we compare C3 versus C4, you can see C4 plants don't have photorespiration. They don't have to fight that. Works only in full sun because you have to have a lot of light input. They use less water. You can see the kind of the differences there. That is something that does come up from time to time on the AP exam, but I'm not directly testing you over it, so that's why I called it Level B. Moving on to CAM, you see what CAM is short for? Crassulation acid metabolism. So yeah, so let's call that CAM. Found in plants where there isn't a lot of water. Things like cacti and succulents and pineapples and orchids and things like that are all going to use CAM photosynthesis. And essentially, because it's so hot, they're going to open their stomata, or their where they absorb CO2 and where they exchange gases. Excuse me, I'm so sorry. So they're going to take in that CO2 and they're going to store it as a C4 acid. So C4 like 4-carbon acid. So that's a more long-term storage molecule than CO2. CO2 would be active in chemical reactions inside the plant. This C4 is not active. So during the day, the acid is going to be gradually broken down and the carbon dioxide is going to be fixed into sugar, and it's going to jumpstart the whole process from that carbon fixation forward. Again, it's not going to use rubisco. It's going to use a different enzyme, much like the C4 plants. So that's the big difference between C3 photosynthesis and then the modifications is the enzyme that you're using. So think about this. If you're storing acid and gradually breaking it down, that's going to mess with your pH throughout the day and throughout the night. So that's going to be something that plant is going to have to be able to withstand. Low pHs during part of the day and high pHs during another part of the day. Sounds stressful to me. So these CAM plants can avoid water stress, or they do avoid water stress, because they keep these stomates closed during the day. They don't have to worry about water evaporating through the holes in their leaves because the leaves are closed. But they're going to have slow growth because they're spending so much of their time and energy storing and then unleashing energy. So just a couple of quick notes here at the end on ratios. If photosynthesis, so PS is photosynthesis, RS is respiration. So respiration is always happening. Photosynthesis doesn't always happen because the plant doesn't always have light. But if photosynthesis is happening more than respiration, the plant can grow and reproduce. If they're exactly equal, their rates, there's not going to be any growth, but it's not going to die either. It's going to be tough for the plant to reproduce at that point as well. And then once respiration outweighs photosynthesis, the plant's going to starve because it's eating more than it's producing. Again, respiration happens 24 hours a day. Photosynthesis only in the light. Plants overwinter on stored food. So during the summer, there's a lot of photosynthesis happening. Once those leaves break down, a lot less is happening. So the best way to increase the photosynthetic rate is to increase light. These things impact photosynthesis, probably nothing surprise there. Importances of photosynthesis, these hopefully are review for you. If they're not, look them up on your own. And here's our summary. Know the balanced equation, know what the dark and light reactions are, know where they happen, know what they produce, what they take in, all that stuff. Know the alternate forms of photosynthesis, so C4 and CAM plants, and then know those comments about photosynthetic and respiration ratios. I did post a practice question on this lecture file on Canvas, so that's a common free response type question. And there are also several links posted on the light reaction in Mr. Anderson's video, all on Canvas on our class page. So that's chapter 10. Let me know if you have any questions.